you, Jeannie. Greetings from Oakland, California, where we're having an influenza outbreak. We've had both influenza and norovirus in my household in the past couple of weeks, but here I am, a survivor, and I do strongly recommend flu shots. Jeannie shared with me a list of the participants, and I see that most of you are TB program people or clinicians or public health nurses, so I'm going to try not to overwhelm you with acronyms or try to explain any lab terminology that you may not be familiar with. Hope I remember to do that. All right, so let's get started. One of the things I want to share with you is the complexities and challenges that we have so that you'll understand that there are limitations to the testing and appreciate how those limitations could guide therapeutic decisions or TB control decisions that you have to make. So, exploding head zone. This was a title that was concocted by somebody else, but really does kind of reflect the current situation. Molecular and growth-based drug susceptibility testing. So, there's good news and bad news. And the good news is that new technologies have improved our turnaround times and accuracy. When I first started working in the TB lab 26 years ago, our turnaround time from a sputum arriving in the lab to the drug susceptibility test going out was about 6 weeks. And now with the molecular methods, we can sometimes turn it around in a day. So we're doing reference drug susceptibility testing by culture-based and molecular methods. This is actually 15 states that have low test volumes, so it's really not cost effective for them to maintain the TB drug susceptibility testing service. The bad news, our new knowledge has shown that our previous understanding of drug susceptibility testing was naive, and so we have a lot of investigating to do and really try to establish the best testing methods that will give the most accurate, rapid results. And the other bad news, which is not new news, but we have inconsistencies in media and techniques that can lead to inconsistent or conflicting results. Okay, so the drug susceptibility testing, DST services at California state labs or other states are funded by CDC through APHL. And so the lower volumes of TB drug susceptibility testing, that means less than 50 a year. So those labs are eligible to participate in this program, and CDC funds us to do it, and we don't charge them. And there's a list of the states for which we're doing work right now. Not all of the states send us all of their work, so there's some states that are signed up but don't fully participate. And this includes us doing routine first-line drug susceptibility testing by the Mycobacteria Growth Indicator Tube, MGIT, Mycobacteria Growth Indicator Tube. So that's INH, rifampin, ethambutol, pyrazinamide, the first-line drugs. When you submit a culture to us, we'll test for susceptibility to those drugs and MGIT. And we will reflex to second-line DST for resistant isolates or on request. The other thing that we do is molecular detection of resistance to INH and rifampin directly on smear-positive specimens with prior approval. And I'll talk about the prior approval in just a minute here. But all rifampin-resistant strains are sent to CDC, their molecular detection of drug resistance service for sequencing. I'm really happy that CDC is thoroughly studying rifampin resistance and the molecular basis for it, and hopefully they'll come up with a good understanding of rifampin resistance in the United States. So when there is a suspicion of drug resistance, that's when you'd want to request pyrosequencing, which is the rapid molecular one-day method. And I think you probably understand when to suspect drug resistance if the patient is coming from some countries where there's a high prevalence of drug resistance, or if they've been previously treated for TB and now they're culture positive again or smear positive, or if they've been exposed to a drug-resistant case, and so forth. So when we get a culture in our laboratory for drug susceptibility testing, the first thing we do, hopefully on the first day, is make a subculture, and we do the drug susceptibility testing on the subculture. So the range of turnaround time from the time we get a culture till the time the subculture grows and we've done the DST, range of 10 to 20 days. And Grace Lynn and our laboratory and our crew have worked real hard to get the mean down to 15 days. But you have to remember that drug-resistant strains may grow more slowly. So I wanted to give you an example of what happens when you don't tell us that you suspect drug resistance. So what happens is the routine thing, which takes a little while. And so this is a case from 2015 when we received an aliquot of MGIT, or midget broth. We subcultured it on the day that we got it, and it grew up about 2 weeks later. It took the full 2 weeks for it to grow. That's because it was a drug-resistant strain. It grew rather slowly. So we first observed growth in the subculture after 2 weeks and set up the first-line drugs. And then 10 days after that, the drug susceptibility test was completed for first-line drugs, and it was MDR, multi-drug resistant. It was resistant to INH and rifampin. When we saw that, we went to the molecular testing, the pyrosequencing, and this indicated resistance to fluoroquinolone and injectable drugs, so moxifloxacin, amikacin. So unfortunately, the turnaround time was 25 days from the time that midget broth arrived in our laboratory. Now, if the submitting lab had told us that they were suspecting drug resistance, we would have done the pyrosequencing immediately, and we would have gotten the molecular results indicating XDR-TB within 1 day instead of 25 days. So please let us know if you're suspecting drug resistance so that we can do the molecular testing as well as the culture-based testing and get the result for you much more quickly instead of the patient coughing up XDR-TB for an additional 24 days. All right, so here's the exploding head graphic, molecular and culture-based DST. I'm hoping that by the end of this talk, you'll understand the complexities. I hope your head doesn't actually explode, but you'll understand that the laboratory testing is not perfect, and sometimes we need to do a combination of different testing methods to arrive at the best results. Okay, so here we are, ethambutol in horror font, and there we have a picture. What do those pictures mean? Okay, on the one on the left means that our gold standard has been tarnished, what we thought was the gold standard method for ethambutol. And the one on the right means that we are now lost in a fog. So I want to share with you some particulars about that. Okay, now this is a slide. It's a pretty busy slide, but there's just a couple of points that I want to make from this. And this is from the expanded MPEP. So the expanded Model Performance Evaluation Program. The Centers for Disease Control sent out a set of 30 TB strains of interest to a multitude of laboratories and had us all do the drug susceptibility testing for the drugs we test using the methods that we use. And among the strains that they sent out, they sent some that had wild type sequence in the EMBB gene. Now the EMBB gene is the one that is sequenced to detect suspected ethambutol resistance. So very often when a strain of TB is resistant to ethambutol, it will have a mutation in the EMBB sequence and won't be wild type. But for these 9 strains, they had the wild type sequence. So based on the sequencing, you would expect them to be susceptible. But you can see that of these 9, most of them were resistant. Actually, 5 of them were resistant by the Auger proportion method in laboratory A. So the point here is that not all ethambutol resistance is detected by EMBB sequencing. You may notice that there are some differences between the different laboratories and the different methods in the results that they obtained. But we'll talk more about that in a minute. So the other point I want to share with you is that MGIT detects less ethambutol resistance than the classical reference agar proportion method. And in another set of cultures that was sent out by CDC in the Model Performance Evaluation Program, this was in November 2010, they sent out duplicate isolates with an EMBB mutation at codon 6, the MET306 val mutation, and 90% of laboratories that tested them using the agar proportion method got a resistant result. And only 23% of the labs using MIDGET detected drug resistance. So, I think for most cultures, the ethambutol results by agar proportion and MIDGET will agree. The frequency of discrepancies is really unknown, but there is a tendency for agar proportion to detect more resistance than MIDGET. Now, we are not sure of the clinical relevance, and we're not sure which of them is correct, but agar proportion is more sensitive in detecting ethambutol resistance. All right, now we have a set of 8 cultures. This is from the 30 cultures that were sent out in the expanded MPEP set. And there were 8 cultures that had the same mutation in EMBB. They all had a mutation at codon 306, the MET306 isoleucine mutation. And in laboratory A, of these, 7 of the 8 tested resistant, but notice in another laboratory, which has excellent quality control, some of these tested susceptible. And if you take a look on the right, for the laboratory that did quantitative testing, MIC testing, that was, by the way, our laboratory, Laboratory C, even though they all had the same mutation in the EMBB gene, there was a pretty big variety of MICs. So the 2 at the bottom, for example, strains 29 and 30, they had a low MIC, less than or equal to 3 micrograms per milliliter. And others had an MIC of greater than 5. So there's a pretty big variety. So isolates with the same mutation may differ widely in susceptibility versus resistance. And the other point from these data is that labs with good quality control, doing agar proportion DST for ethambutol, may differ significantly in results. So what are the findings from the MPEP and the expanded MPEP regarding ethambutol? Some strains resistant to ethambutol do not have mutations in the EMBB region, which is routinely sequenced. And we know that laboratories with strong QC programs, quality control, may differ from each other in their agar proportion ethambutol results with the same strains. We found that TB strains with the same EMBB mutation may differ significantly in their ethambutol MICs. And we found that MIDGET may detect less resistance than agar proportion. So is your head ready to explode? All right, so on what can we rely? So I would like to say that patient isolates that are susceptible to isoniazid are unlikely to be resistant to ethambutol. And I want to refer here to a paper from 2002. Now, in this paper from 2002, you'll notice that the technique that was used here was different from the technique we use now. The technique that we used is that was used back in 2002 is the BACTEC 12B medium. And for the BACTEC 12B medium, this had a test concentration that was probably a little bit too low, and it tended to yield false resistance. Nowadays, we're using a different medium, which is the MGIT, and I think that maybe the test concentration in MGIT is a little too high, so it tends to yield some false susceptibility. But I think in this 2002 study, it clearly showed one thing, that strains which are truly resistant to ethambutol, 97% of the time, they're also resistant to isoniazid. So for isolates which test susceptible to both INH and ethambutol, the ethambutol result can be considered reliable, in my opinion. Okay. What about INH resistant strains? Is a midget susceptible result for ethambutol reliable? Some strains, for example, the ones with the MET306 val mutation, may test susceptible in midget and resistant by agar proportion. So for evaluating the treatment efficacy, it's unclear which one is correct. One way to arrive at a decision, test by both molecular and culture-based methods and synthesize two results. And conflicting results may indicate diminished susceptibility, but ethambutol might still be a useful part of the regimen. So more about synthesizing the ethambutol results. If you've got, in my opinion, a result of INH susceptible and ethambutol susceptible, that's very likely going to be ethambutol susceptible. INH susceptible and ethambutol resistant, that is a questionable result. That's going to occur so rarely that it should be repeated. Unusual laboratory results should have repeat testing. And if you've got INH resistant and ethambutol resistant by any method, agar or midget or sequencing, this should tell you that it's not fully susceptible to ethambutol and likely resistant to ethambutol. And if you've got ethambutol susceptible by midget and resistant by agar, I guess it's not fully susceptible, but the clinical relevance is unknown. So we have sort of a de facto intermediate or gray zone here if this result occurs of ethambutol susceptible in midget and resistant by agar. So here's some ethambutol research questions to lead us out of this fog. TB strains with wild-type sequences in EMB-B gene may have high EMB MICs. So the research question is, what other genes and mutations are associated with resistance? Another question, TB strains with the same EMB-B mutation may have significantly different MICs. So how do combinations of mutations affect susceptibility or resistance quantitatively? So the linking of whole genome sequencing data with MIC values may provide answers in the future. It's going to take a lot of work. Some more research questions about EMB, testing in midget in particular. Where is the clinical correlation? We can work to make different laboratory methods agree, but are we really doing a better job of predicting treatment success or failure? I would like to suggest that a rodent study might help. So for the rodent study, we would get a set of strains with a range of different EMB ethambutol MICs, quantitative drug susceptibility results. And we would infect rodents with these strains and then treat with ethambutol at the doses equivalent to human therapy. And then following treatment, we would sacrifice the mice and evaluate whether ethambutol had been effective in inhibiting growth in the lungs of the mice. Then we could establish an MIC cut point above which ethambutol treatment is not likely to be effective. Okay, so for ethambutol, we had the tarnished gold and the ship lost in the fog. And here for pyrazinamide, we've got a somewhat crazed-looking rabbit. Crazy doesn't even begin to cover it. Pyrazinamide, some of the problems we've known for a long time. Here's an article by Chador from 2010 and other people. 57 isolates that tested resistant to PZA in her laboratory in midget, only 58% of them were resistant on repeat testing. So unverifiable, unrepeatable resistance to PZA commonly occurs. And I think this is why Ying Zhang from Johns Hopkins published in 2014 that current phenotype-based susceptibility testing is not reliable due to false resistance. Sequencing of the PNCA gene, which I'm going to call pink A, it's a pyrazinamidase gene, but the pink A gene represents a more rapid cost-effective and reliable molecular tests for PZA susceptibility testing. So here's the challenges of testing pyrazinamide in microbacteria growth indicator tube in midget. If the inoculum is too heavy, the pH may be raised, and at a raised pH, drug activity will decrease, and then you're going to have false resistance. If the inoculum is too light, it may not grow because PZA medium is a low pH medium, which is not really ideal for growing TB. So if the inoculum is too light, you may get false susceptibility. And if the culture being inoculated is old, the metabolically inactive cells may have increased susceptibility to PZA. So it's very difficult to get it right. And this is described in an earlier article by Ying Zhang from 2003. So a proposal for pyrazinamide. Use fresh cultures for inoculation to avoid false susceptibility. In bold here, avoid over-inoculation to avoid false resistance. When a culture tests pyrazinamide monoresistant, repeat the test and investigate. So the investigation should include, you want to think about whether the strain might be Mycobacterium bovis, and bovis is more commonly isolated from extrapulmonary sites, or do you have some other reason to suspect bovis? So if it's bovis, this would support that the pyrazinamide resistant result is correct, because bovis strains are inherently pyrazinamide resistant. So when repeat retesting result becomes available, if it's resistant again, so it's confirmed, you can report resistance. Now, if it's susceptible, ideally we would like to sequence the pink A gene, including the promoter region. And I don't know how readily accessible this pink A sequencing is going to be. I'm not volunteering the CDC laboratory to test all of the discrepant strains that had a resistant result initially and a susceptible result on retest, because that might overburden their laboratory. But this would be an ideal way to address the discrepancy. So the molecular basis of resistance to pyrazinamide from a Ying Zhang article from 2015, mutations in the pink A gene are the main mechanism of resistance. The mutations in this gene are highly diverse and scattered along the gene, which is unique to PZA resistance, that doesn't have hot spots. Now here it says most PZA-resistant TB strains, 72 to 99%, have mutations in pink A, but some do not. Now in some of these cases where you have a resistant TB strain in culture, but there's no mutation in pink A, the culture-based DST is notoriously unreliable. So maybe the pink A sequence was correct. So I would tend to be more inclined to think that a higher percentage of PZA-resistant strains have pink A mutations. But it's really difficult to verify. There are a couple of other gene loci, RPSA and PAN-D, that have been associated with PZA resistance. We don't really know at the moment how frequently these are associated with resistance to pyrazinamide, because really most of it may be due to mutations in the pink A gene. All right, now going to a different subject. Lot-to-lot variability in culture media sometimes affects drug susceptibility testing results. And there was a publication from our laboratory, the Microbial Diseases Laboratory, in 1988 by Linda Guthertz and others. This is probably the most read and most quoted article on TB from our laboratory. So here's something from the conclusion. This study demonstrates that individual lots of components of the basal medium may vary significantly in their suitability for susceptibility testing, and failure to detect such variation may dramatically affect susceptibility profiles. So when we're doing drug susceptibility testing, we've got a medium base and a growth supplement, the oleic acid albumin dextrose catalase. They contain biological materials, like, for example, the albumin, which may vary from lot to lot. So this may affect growth support or drug activity in media made with these components. So picture's worth 1,000 words. So this is from the 1988 article. So these are inoculation of two drug susceptibility testing cultures with the same strain. And in the quadrant number 1 up here at 12 o'clock, both cultures were inoculated with the same inoculum. Now here on the right, this is a new batch of medium made with a new batch of the growth supplement. And you'll notice that the growth is much less luxurious. So the new growth supplement was not as good at supporting growth as the reference lot of medium. So what effect did this have on drug susceptibility testing? You notice in this quadrant here at 3 o'clock, they're resistant in both cases, and here down at 6 o'clock, there's resistance in both. But take a look here at the quadrant at 9 o'clock. In the original medium, okay, thank you, Jeannie. Okay, so in the original medium here, there were some colonies in this 9 o'clock, this quadrant at 9 o'clock, whereas here in the medium, which had a growth supplement that was not as good at supporting growth, there were no colonies here. So you might conclude from this test batch that this strain was susceptible to this drug, which I think might have been streptomycin here in this quadrant, that it was susceptible. It's not really susceptible. It's just that the medium is less good at supporting growth. So the converse can happen as well. From the same paper, the growth quadrant with no drug is at the upper right here. And you can see that both the old on the left and the new on the right, the old medium and the new medium both had similarly luxuriant growth in the control quadrant. But take a look here down in this quadrant right around 8 o'clock. In the old, the reference lot of medium, growth was inhibited by this drug. And in the new batch of medium, whoa, there was growth. What seems to have happened is that the growth supplement or the basal medium in the new batch of medium bound and inactivated the drug so that you got a false impression of resistance. So that can happen too. A new batch of medium can fail to support growth, or it can bind and inactivate some of the drug. So does variability in culture-based DST systems make them less reliable? Now here's some data from Grace Lynn in our laboratory with pyrazinamide. And you'll notice that in the testing that we did, we tested the laboratory stock strain, H37RV, and routinely, for a long period of time, the MIC that we got for H37RV was 25 micrograms per mil. That's the lowest concentration of drug that would inhibit this control strain. But in August 2014, the MIC changed. The MIC became a little higher. That's not a big variation, a single dilution, a twofold dilution. But in April 2015, when we did quality control testing, whoops, I'm pushing the wrong thing. The MIC went up to 100. So the quality control MIC testing of the stock strain indicated that there may have been a change in the PZA medium for drug susceptibility testing, which we bought from Becton Dickinson. Okay. So lot-to-lot variability. What does it say in the Clinical Laboratory Standard Institute standard, M24, for mycobacterial drug susceptibility testing. It says laboratories should choose a broth-based system from a manufacturer that performs regular quality control and quality assurance testing to ensure that the median drug performance are consistent from one lot to the next, and that the median drug lots provide consistent results over long periods of time, for example, years, without drift. And further, it says when deciding which shorter incubation broth system to use, it's reasonable to ask the manufacturer to describe the measures they take to ensure lot-to-lot consistency in the drug susceptibility results, and to consider this information in deciding which system to use. Now, both manufacturers of the rapid broth testing systems, Midget and Versatrek, they essentially abnegate this responsibility. They do not test MICs the way we do in our laboratory to make sure that they have a consistent MIC from lot to lot and year after year. So what are we going to do about this? One approach, we can hope that the manufacturer will define the medium components chemically as carefully as possible and do pre-market chemical testing of the composition of these components to assure lot-to-lot consistency. But they have to be very careful to evaluate the ingredients and do careful chemical testing. And the other approach is when we in the laboratory get a new batch of medium components, we make up some media with the new and some of the old components, and we compare growth support and drug MICs between new and old lots. And I can tell you this is an onerous responsibility, but we do this in our laboratory. So some take-home points here. DNA sequencing provides rapid results that are not altered by subtle changes in reagents. But the correlation between sequence change and drug susceptibility may be complex and multifactorial for some drugs. And elucidation of this may require extensive study over decades, probably. Although some mutations are clearly associated with resistance, we've learned that culture-based drug susceptibility testing may be affected by subtle changes in culture medium ingredients. And is rigorous quality assurance by the end user, like MIC testing, practical? I sure wish the manufacturers would do it instead. But all they seem to be willing to do is pre-market testing for the chemical composition of the medium. Another take-home point, I think that for ethambutol, the clinical correlations of DST may need to be reestablished. And we may be able in the future to design testing algorithms to overcome the weaknesses in the current test methods, perhaps an algorithm that uses both molecular and culture-based testing. Okay. Here's some good news. Cepheid is coming out with Gene Expert MTB-RIF Ultra, expected to come out soon, maybe this year. And it's expected to have a greater sensitivity with smear-negative specimens. This makes some sense because you'll notice that among the targets in this real-time PCR system is IS6110. This is a gene segment which can be present in multiple copies. The earlier generation of Gene Expert was looking for RPOB gene, which probably is present only in one copy in the cell. And if you're looking for a gene segment that's present in multiple copies, you'd expect to have better sensitivity. And indeed, the sensitivity with smear-negative samples is reported as 90%. This is improved over the 70% or so, which was found with smear-negative samples in the earlier Gene Expert product. Another Cepheid product that may be coming out or may not is an XDRTB product. The chemistry has been established for it, the molecular beacons with melting curves, which can determine resistance not only to rifampin, but also isoniazid and fluoroquinolones and injectables. So if this product becomes available, it's going to provide more information than the MTB-RIF product from Cepheid, which is the one that's available now. Okay, so why do the Association of Public Health Labs and CDC recommend DNA sequencing each time the Cepheid gene expert detects an RPOB mutation? So this is the current generation of product with a real-time PCR that's available commercially and cleared by the U.S. Food and Drug Administration. In DNA sequencing performed at the CDC laboratory and also at the Microbial Diseases Lab, our laboratory in Richmond, California, 18 to 20% of the mutations found by GeneXpert are silent or synonymous mutations, which don't confer resistance. So in much of the literature that you read, the studies on GeneXpert were done in high-burden countries where true resistance to rifampin is much more prevalent. So globally, the incidence of MDR-TB is about 5 times higher than it is in the USA. So they have a lot more true resistance, and so the silent mutations are much less concern, much lower percentage in the places where the first studies were done about the accuracy of GeneXpert. And here are some data from the Texas State Laboratory. I'd like to give a nod here to Ken Jost from Texas, who's provided these data and is about to retire after a very distinguished career. Thank you so much, Ken, for this contribution and so many others. But at the Texas Laboratory, over a period of a couple of years, they tested 434 specimens. And you'll notice that in the GeneXpert, they had 15 that tested resistant to rifampin by the GeneXpert. And when they did the culture-based drug susceptibility testing, 9 of them were resistant, and 6 of them were susceptible. So the predictive value of a resistant result for rifampin by GeneXpert was 60%. That's a little disappointing. So they undertook DNA sequencing of the RPOB gene, and they found out that there was one particular probe, and the GeneXpert has 5 probes, A through E, but mutations in the B probe were not really associated with true rifampin resistance. In this study, many of the mutations detected by the B probe were silent mutations. So you can keep that in mind when the GeneXpert has detected a mutation in the RPOB gene. If that mutation is detected by probe B, this is much less predictive of rifampin resistance. And DNA sequencing will help you to find out quickly whether you can expect drug resistance to rifampin. And also culture-based drug susceptibility testing should always be done. So thanks again to Ken. So some RPOB mutations are clinically significant. This is a different issue. This is an issue about the low-level rifampin resistance, which I want to take on next. But there are some strains that have an RPOB mutation, and they may test susceptible in midget. So let's, for the purpose of this talk, let's define low-level rifampin resistance to be the presence of a mutation in the RPOB gene, which causes a change in the amino acid sequence, leading to an increase in MIC, mineral inhibitory concentration, above that of the wild type, but less than 1 microgram per mil in midget, so that the culture tests susceptible in midget. So that's low-level rifampin resistance. And a study in New Zealand, published in 2012, found that for strains that have these mutations, treatment failed for these patients. For these 3 patients, they had mutations at 526 and the 516 loci. They tested susceptible in midget, and the patient's failed therapy. And we have a study from our laboratory, which I'll talk about in a minute here, led by Neha Shah, which found essentially the same thing, that these mutations are associated with treatment failure. But greater use of the gene expert MTB-RIF, detecting sequence changes, could increase discovery of low-level resistance. If the gene expert says resistant and the midget says susceptible, it should be sent for DNA sequencing, and then we can find out if it's one of these low-level resistance mutations. So expanded use of gene expert or some other system which detects mutations in RPOB gene is recommended. And when there is low-level resistance, for example, there's a midget expert discrepancy or difference detected by sequencing, the treatment regimen may need to be modified, which is the finding of Dr. Shah from our lab. Okay, so a low-level rifampin resistance recap. If there's an RPOB mutation and it's also resistant in midget, then you can say this is clearly rifampin resistant and rifampin is not useful. With low-level resistance, you've got an RPOB mutation. It might test susceptible in midget, but if it's not a silent mutation, expert consultation may be needed because treatment may fail. And if DNA sequencing is done, we may be able to help you know whether the mutation, the specific mutation that's present, has been associated with treatment failure. But if there's no RPOB mutation and it tests susceptible by midget, then it's reliably going to be susceptible to rifampin. Okay, here's some other interesting findings, again from data from Grace Lynn and other people in our laboratory. And they took a look at the most common mutations associated with resistance to rifampin. So you notice the most common mutation is this 531 mutation, TCG to TTG. It's associated with high rifampin MIC. It's very resistant to rifampin. And here, the numbers here in black are the MIC to a different but related drug, rifabutin. So this strains with this 531 mutation, they're very resistant to rifampin. They're also very resistant to rifabutin. They have high MICs to rifabutin. But take a look, on the other hand, at this fairly commonly occurring mutation. We found 17 of these, about half the number of the 531 TTG. They're also very resistant to rifampin, but take a look at the rifabutin MICs. They're rather low. They're in the range of 0.25 to 0.5. So rifabutin may have some activity and may be useful in treating these strains. And there's a number of different mutations that are similarly associated with this, mutations in the 526 gene, 526 locus, for example, may be associated with rifampin resistance, but a low MIC to rifabutin. So if you get an expert consultation, your consultant may recommend adding rifabutin to the regimen, treatment regimen. How much it's going to be relied on, I don't know, but it may be useful. Okay, we've got some new drugs like bedaquiline. So how do we establish test concentrations for drug susceptibility testing? So we have the classic Canetti discrimination method. So this is the method on which we rely, published way back in 1969. So for the discrimination method, we test the MICs of strains from untreated patients, wild-type strains, and then we also test the MICs, minimal inhibitory concentration of strains from treatment failures. And our test concentration in the laboratory is the concentration at which the wild-type strains are susceptible and the clinically resistant strains are resistant. So it really doesn't matter what other drugs the patient was treated with. If a patient is still culture positive after a regimen of treatment with the drug that we're interested in, the isolate did not respond to that drug, and it's clinically resistant. So clinically resistant strains may be different from resistant strains that are generated in the test tube because they have to survive in human tissue in the presence of an immune system. So unfortunately, we can't just generate the resistant strains in the lab in a test tube. And it certainly can be challenging to obtain TB strains that are clinically resistant to a new drug. It may take us a while to do that. So what information can we obtain from clinically resistant strains? We can find out what mutations they have. This is particularly useful if you have a strain from a specimen that was collected from a patient before therapy, and then the clinically resistant strain from the same patient if therapy fails. So mutations found in this case are likely to be associated with drug resistance. And for the clinically resistant strains, we want to find out what are their minimal inhibitory concentrations and how do those compare with the MICs of wild-type strains. One of the things you have to be careful about is to make sure that the wild-type strains have not been treated with a related drug, because that can cause selection of a mutation. And so we think that the wild-type strain should be pan-susceptible strains. That means susceptible to all the drugs, no resistance. And these are most likely to be truly wild-type. Okay. But when we've got a new drug, it's going to take a while for us to collect the clinically resistant strains. So what can we do if we have few or no clinically resistant strains? We can evaluate the MIC range of wild-type strains, and isolates with MICs higher than this are probably resistant. But where to set the critical concentration? Are we going to set it just above the upper limit of MICs of the wild strains? If we do that, then some variability in testing could lead to false resistance. Or should we set it fourfold higher than the upper limit of MICs of wild strains? But if clinically resistant strains have MICs that are lower than this, then the false susceptible results are possible. I've read a proposal of the concept of using an epidemiologic cutoff. I think you're going to discover by the time I'm finished talking about this that I'm not in favor, but it was published in the Bulletin of the World Health Organization. So what they propose is that the highest MIC within the wild-type MIC distribution should be called the epidemiologic cutoff. And then they use the epidemiologic cutoff together with clinical and pharmacokinetic data to make decisions about test concentrations. So they're recommending that susceptibility breakpoints use the same tools that are used for other bacteria. In other words, they don't want to be bothered with the chore of having to collect strains from treatment failure cases and seeing what their MICs are. So this inspires me to quote George Santayana: "Those who do not remember the past are condemned to repeat it." And I think we could run into trouble if we try to use the ECOF, or epidemiologic cutoff. Their proposal was that susceptibility breakpoint should be based on the same tools that's used for other bacteria and fungi, but in tuberculosis, some bacteria are intracellular, some are extracellular, so the usual pharmacokinetic studies don't apply, and some TB bacilli are metabolically dormant, and some of them are actively growing, so the drugs concentrations need to kill these varying populations may differ. So I want to quote the paper by Bill Berman from 1997, the paper that I have read many times and probably the most influential study I've read in my life. the value of in vitro drug activity and pharmacokinetics in predicting the effectiveness of antimicrobacterial therapy, a critical review. A few years ago, I asked Bill Berman to repeat this study, and he's far too busy. He's head of Denver Public Health now. But quoting from his article, in vitro measures of drug activity do not predict the sterilizing activity of a drug or the activity of drug combinations. In vitro measures of drug activity do not allow reliable predictions of the level at which an organism should be considered resistant. And assays of drug penetration in tissues and activity against intracellular bacilli add modestly to the predictive value of in vitro measures, but still do not predict sterilizing activity. So how do we decide on test concentrations for TB drug susceptibility testing? So on the shoulders of giants, we can see a long way. We have the Kennedy discrimination method, which requires collecting clinically resistant strains, and this should enable us to reliably set test concentrations for the laboratory. So I think that's my last slide. Here's our group. I should point out that Grace Lynn, whose name I've mentioned several times, is here. Oh, whoops. So this is Grace Lynn right here. I'll get this eventually. I'm pushing the wrong button. Here we go. All right. Eventually, I'm going to point to Grace Lynn. Okay, here she is. All right, that's Grace Lynn there, the distinguished-looking lady here with the gray hair. And this is our TB lab supervisor, Yvonne Zhang, and so this is our TB laboratory group. And I thank you very much for your attention.